Welcome, everyone. Welcome to GTC. My name is Jason Fenwick. I'm on the Genomics and Healthcare team here at NVIDIA, and today I'll be the host for the session. Our speaker for today is Jurgi Camblong. He is pioneering the global data-driven medicine movement, co-founded SOPHiA GENETICS in 2011, is a member of the Advisory Council on Digital Transformation for the Swiss government, and was recognized by EY as Emergent Entrepreneur of the Year. A molecular biologist with a PhD and EMBA, he has been published in Cell Science and Nature. Please join me in welcoming to the stage Jurgi Camblong.
Wow, thank you very much, Jason. I wasn't expecting to see so many people, so now the expectations are high, and I hope I'm going to be able to inspire you. I just arrived from Switzerland last evening, so my energy level should be pretty good this hour. So you've heard a lot about AI in many disciplines. In healthcare, it's something that will be very important, but today AI in healthcare is really lagging for diverse reasons. And I'm going to try to explain you, despite that, why AI in healthcare is so important. So let me first start by one trend, which is not a very positive trend, unfortunately, is how over years, the number of cancer cases and the diagnosis of cancer cases is ramping up. Today, it's about 20 million patients that are on a yearly basis diagnosed cancer. And we should expect, with the aging of the population, those numbers to continue rise. On the other side, and we don't necessarily know exactly why, but there are more and more as well rare inherited disorders. A rare inherited disorder is a disorder that is present in less than 1 person out of 1,000 patients. So definitively in this room, there are people who suffer from a rare inherited disorder. It's about 6% of the population that suffers from that. So while these are two trends that we cannot change, we have to try to make something out of it to better diagnose those patients and eventually have a better idea of what will be the prognosis as well as how we're going to treat those patients. Now, the good news is that over the last 20 years, there have been a number of new technologies and modalities that arised, and in particular, a data modality that has become very important called genomics. And that thanks to this data, now we are in a much better position to understand what is the origin of this type of disorders. Actually, I don't know if you remember, but the first time ever the human genome was sequenced was only in 2003. So it's quite recent, right? And it took $3 billion and 30 years to make it happen. And now in many labs around the world, you have equipments called next-generation DNA sequencers that actually enable the deciphering, the diagnosis of patients through the production of the genomics information. Beyond that, there are other data modalities, such as imaging, digital pathology, that are becoming more and more important, more and more digitalized, more and more complex as well. And it is estimated that it's about 30% of the world data that are being produced today, 30%, 30, that are health data. So massive need, massive amount of data, and hence the opportunity of applying smart algorithms, artificial intelligence, to be able to leverage on those data, and by leveraging on those data to make discoveries, better understand biology, but as well help patients around the world. So my next slide will be a bit ironic. So for the one who come from the tech world, please don't beat me up. But my message is AI has been applied to many things, okay? And obviously, most of us have cars, maybe not all of us anymore. But on self-driving cars, we knew the physics. This was a world we created as human beings. We knew the cars, we knew the roads, we know the turns. There is some uncertainty, but the level of uncertainty, it's very limited, okay? So now try to think about a world where we're being training cars to drive by themselves, but that where the physical foundations of the world may change. The governing foundations that we wouldn't necessarily master, no. I don't know if here, gravity would help or not. I'm not a physician, but it would be very hard to have a car that has been trained on our roads evolve in a proper way when the world is changing in a way that we have never anticipated, right? I guess you agree on that? Yes? So look, that's the biggest challenge in biology. Biology is quite new discipline. We know the structure of the DNA only since 1950-something. And actually, thanks to physics, to chemistry, and so on, we've been becoming better and better at understanding biology. But we still realize that there are a lot of things that we don't understand in biology. And biology tends to be something which is quite stochastic. So for example, if you think about cancer, it might be that the cells are developing in a certain way and may jump completely to another dimension, a bit like if an electron is moving from one circle to the other. And just to think about the number of possibilities and reactions that are happening in a cell is millions of molecular reactions that are being made every second. It's DNA repair, DNA replication, DNA to RNA transcription, protein modifications, protein synthesis, and beyond that as well, a lot of metabolic reactions. And that only one single cell. We are made of billions of cells, right? So think about the complexity that this is being creating. So building AI in general for biology is very hard, and building AI for healthcare is very hard as well. Now, these are the three main things that, at least from our experience, have proven to be very, very important to be able to build proper AI models that are not solving everything. As I said, biology is far too complex. We haven't built it, but at least at being able to bring benefits to patients. One is a deep understanding of biology. And in my experience, this has been often underestimated. It's not just a data compute problem. It's really understanding how the data are being produced and what is the biology behind it. Otherwise, it's very, very hard to correct the signals and get the signal out of the noise. The second one, which is not unique to healthcare, and obviously a lot of people are talking about that in the context of LLMs, is access to vast amount of diverse data sets. But in healthcare, given the complexity, it's even more important to be exposed to this diversity. And the third one is the ability of leveraging on AI algorithms to be able to get something out of this data and help patients around the world. Now, while the one and the two, the biological understanding and the understanding on building algorithms are, we could say, technical, the one in the middle, it's not technical, actually, and it's something that is very, very hard. And I don't know how much awareness you have around that, but our healthcare data, your healthcare data are being trapped into institutions, okay? And maybe at the beginning, they are being trapped for good reasons. Could be fear that my data might be used against myself with insurance. Could be so that researchers can be more competitive in one institution versus the other. But actually understanding technology and AI here at NVIDIA GTC, I think we all agree that being exposed to these diverse data sets could enable us to build algorithms that are more powerful. Today, it's very, very rare to see institutions that are sharing data across radiologists, pathologists, and oncologists. One good example, actually, is a partner of us in New York, Memorial Sloan Kettering, who has a huge database of patients where they have all the data modalities. But beyond that, even at MSK, today they are not, I would say, day in, day out collaborating with other institutions to leverage on this data, right? Hence the need of unlocking the power of this data wisely, respecting data privacy, which means respecting the wish of a patient. Do I want or I don't want my data to be leveraged? To build a world where everyone benefits from a collective intelligence. And so ourselves, when we started in 2011 with no technology, just a dream and our experience in what we call at the time in research systems biology, the idea was how we can animate a community and be part of it so that this data flow, and thanks to the exposure of this data, the algorithms are learning, and ultimately, what we build is not an algorithm, it's not artificial intelligence, it's not technical, it's a collective intelligence. A collective intelligence that enables any doctor to benefit from patient cases from other doctors that have been working around the world, but in a comprehensive way. So for that, in many, many years, we've been working on building a platform, and we actually impose ourselves some principles. First, we decided to be in the cloud, starting in Europe with no product, 2011, cloud, oh la la, this is evil, right? Please, no cloud, we don't trust that. Wasn't easy. But I think was essential because if you think about the ultimate objective, which is creating this collective intelligence, if you are not in the cloud, I don't think you are going very far. The second one was to be tech agnostic. Very hard, and you will see in a minute, I will drive you into the labs, but in the labs, they are using a lot of technologies, and these technologies are being sent by different vendors, and the type of data that are being produced by the different technologies are different, and it's very hard to harmonize them. We decided that despite this was an effort, we would build a technology that would scale for any sequencer, for any imaging instrument. And the third was to do that without agreeing on making exclusivities with an industry vendor that may be selling an instrument or with one single hospital, because otherwise, again, our dream of building this collective intelligence wouldn't work. So on paper, it sounds easy. Practically, when you're running a business, it's not easy because sometimes companies are offering you a lot of money for exclusivity, but you need to resist to the temptation for a longer-term ambition. And thanks to that, we built a platform that has a menu in oncology and rare inherited disorders, where we have AI factories in genomics, radiomics, and multimodal that enable us to extract signal, harmonize that, and you will see some use cases soon. And a data warehouse where now we can leverage on our privacy-preserving principles to as well enable collaborations across institutions. So it's still the beginning, I would say, because I said 20 million patients every year that suffer from cancer and are being diagnosed, and about 6% of the population suffering from rare inherited disorders, but still quite an achievement. Last year, our platform has been used routinely around the world, which, as you may remember, my desire of creating a collective intelligence was important. 70 countries, 800 tier 1 healthcare institutions, to better decipher what is driving the cancer or the rare disorder of over 350,000 patients. So, while this is important, now I'm going to bring you with me into the lab. And before I was an entrepreneur, I was a biologist as well. And so I thought I would bring my lab coat, which I can reassure you I cleaned up before I would come here. And I will explain you why, although it sounds simple, identifying specific mutations of specific patients is not so easy, okay? So it's going to be a little bit of lab, a little bit of biology. I hope that I will make it entertaining and that you're going to be able to follow me. So about 20 years ago, when you would have a lung cancer, we would say it's a lung cancer. Now we know, because of the molecular drivers, that actually there are hundreds of different lung cancers. So one could be a ALK lung cancer, an EGFR lung cancer. So those mutations are defining as well the origin, right? That makes sense. You're not fixing your car because it's a Tesla or because it's a Renault or an Audi in a certain way. You really need to understand what is driving basically the break in your car. Same applies to cancer with the molecular information. And by doing so, as you can see, even you end up then having some cancers which might be breast or ovarian that are both led by BRCA mutations. Okay, so actually same mechanisms that are not working, but on different tissues. Now, identifying these mutations is not easy because in the labs, as I said, what they do is you're going to buy an equipment, which is called the next generation DNA sequencer, which is biology and enzymes inside the device, and is going to produce dirty data very rapidly. It was 30 years in 2003. Now you can do it in a day, but it's very dirty that comes out of it. It's very good because we need that, but it's dirty. The algorithm needs to understand the signal to basically bring eliminate the noise to bring the signal. The second element is before you are sequencing the information and digitalizing in the sequencers, you need to multiply the DNA. All of you, thanks to COVID, know what PCR is about. Same principles apply in labs to amplify basically DNA, not exactly the same technologies, but this is as well biology in a tube and brings a lot of biases into the data. And the last one is a sample type. Depending on the quality of the tissue, of the blood, the quantity of mutations, the signal that you get into a sample is very different. So the challenge is that those things are different from one lab to another, from one hospital to the other, and are bringing noise into what you see the raw genomics data on the right side. And obviously, the combination of these diversities make even the data more complex and less harmonious. So that's why understanding the biology here and what are the penalties that are being brought by these different steps in the lab is extremely important to be able to properly identify mutations. So again, sounds simple. All of you may think that from the sequencer, I have a signal. It's not true. You need AI. You need to understand this process to make it happen. Otherwise, the same sample in different labs will give you a different outcome, which I think we would all acknowledge is not fair. So to solve this problem at SOPHiA, we've been exposed to millions of cases of specific type of mutations where we had to run through, which we call in our jargon the orthogonal assay, another lab technology. And by understanding what may bring signal and noise, we've been building algorithms to be able to harmonize the data and extract proper signal. These are our algorithms. As you can see, they have the names of spices. What we say is that we're adding these spices to make the data a bit more appealing. And today I would like to go a bit deeper in the one in the center, which is called ginger. So now, follow me. I'm going to go a little bit into biology. In our DNA, we can have multiple types of DNA breaks for multiple reasons, and there are multiple ways of repairing that. One type of break is called single-strand DNA break. It means only one of your DNA, I don't know, we say ladder in English maybe, is being broken. Other types of breaks are double-strand break DNAs, okay? So now understanding what is driving these breaks is extremely important because with knowledge, we're being able to build approaches which are therapeutical for a specific type of drugs. On the left side, if you are being, normally you have a single-strand DNA break, this is being recognized by a complex and this is being repaired through polymers with a PARP complex. On the left side, if the PARP doesn't work and it has been inhibited, this will lead to actually now double breaks DNA, which normally are being repaired by a famous pathway which is driven and dominated by the BRCA genes called HRR pathway, homologous recombination repair. Now guess what? This was genius. The idea was sometimes cancer is being driven by a lack of the ability of cells to repair these double breaks, which is because the HRR pathway is inactive. So what if in those type of cancers, we would, in addition to that, block the PARP pathway with an inhibitor and then lead to cancer cell? And so this is something that now has enabled pharma companies to deliver into the market called PARP inhibitors. But it was very hard then once the drug was approved to identify patients that would benefit from that. And here quickly I'm highlighting a methodology that we built ourselves on the basis of a deep learning algorithm, and that was approved, actually that was published in Cell Reports Medicine, where we've been able to, thanks to deep learning technologies, understand if in patient samples this HRR pathway was active or inactive. If defective, it means the patient is ineligible to PARP inhibitors. This leads to what is called synthetic lethality, and cancer dies. And this is how this integrates into the workflow of a lab. So basically, the lab is going to produce sequence information about 40 genes which are associated to normally the cause of not being proficient in homologous recombination repair. And what we develop aside that is a way to investigate the consequence of this defect, which is what we call phenotype, which is the scarf of basically the defect. And to do that, we develop a deep learning algorithm applying convolutional neural network to look at low-profile genome sequencing, and from this low-profile genome sequencing, infer if there are number of mutations that are being accumulated because the HR pathway is not working. Actually, this proved to be very, very efficient, and today is being used monthly to help thousands of patients to be properly diagnosed and being prescribed PARP inhibitors. So now in that same spirit, I'm going to let you have a bit of break of my French accent. And I will share with you the case of one patient in Brazil, Gabriel, who was diagnosed a glioma when he was not even a year, and who, thanks to this ability of properly investigating variants, is still alive. But I will leave the Dr. Gustavo Ramos Teixeira speak about it.
I'd like to share a case of a baby that came to us 4 years ago this baby was referenced because the suspicious of a malignant brain tumor at the time. Image analysis revealed a 5 centimeters tumor in his brain. Our neurosurgery tumor was able to perform a great surgery, and the material came to our laboratory. We analyzed it, and the conclusion was that this patient had a high-grade glioma. That's a high-grade tumor that young patients may have, and these cases are known of having worse, a bad prognosis, sometimes a dismal prognosis, and we felt that this case needed a different approach in terms of genomics. What we decided was to perform an NGS panel, especially for the search of fusions, because there were a few researchers, a few publications at the time saying that these tumors, especially in this age, may have tyrosine kinase alterations, receptor alterations, and we decided to perform an NGS, and the result was we detected a fusion in one specific tyrosine kinase receptor, and the directors in the hospital decided that we should pay for this patient's treatment, targeted therapy, because there was a specific therapy for this kind of alteration. And our main result was that at this moment this patient has 4 years old, it's alive and well, and didn't need different surgeries, didn't need a different approach in terms of chemotherapy, with no neurological disturbance. So we think that this kind of approach in terms of genomics, with the aid of SOPHiA GENETICS, SOPHiA DGM platform, for example, it's what helps us to give a better healthcare for our patients, and we hope they can reproduce this case for all of them. Of course, depends of kind of the tumor, depends of the kind of genomics, but that what we think we can, how we can change this patient's lives.
Being able to understand really what is driving the cancer and applying a specific drug, although off-label, really enable that patient to survive from cancer, at least up to today and hopefully for the future. And obviously this is what we want to do for many patients, but this requires a lot of investment, a lot of precision. So today, I'm very proud to announce that actually on March 15th, which was my birthday, we've been able to hit the 2 million patients that benefited routinely from our platform around the world. $2 million sounds still small, right? But was not easy to make it happen. And we are very, very happy to see the progress, and hopefully in our journey to impact on many, many more patients. But obviously, being able to help on cases of Gabriel now brings us the means as well to educate doctors so that we can help other similar cases around the world. Now, 2 million patient genomic data is a lot of data. It's hundreds of terabytes of data, petabytes of data. And I thought for the ones that are very tech savvy, it would be nice to share with you how that volume of data has been evolving over the years. So actually, in our platform, it to keep your immune system very active so that they can recognize the cancer and kill the cancer. Now, the downside of it is that when you maintain your immune system very active, you're attacking your own body as well. So there is as well toxicity versus the body. But indeed, these drugs have proven to be very, very powerful now that they are being used and combined with other drugs in routine clinical care. The challenge is to try to understand what are the features that are giving us a sense of who is going to respond or not respond well to immunotherapy. And so here, our approach that we have been demonstrated works on thousands of patients, has been first starting with CT scan data, and being able to segment the tumor, which is not easy, because these are metastatic cases. So unlike on early cancer where segmentation can be quite easy, this is delocalized cancer, so you need to identify the different metastases in the body. Then, once this has been done, extract signal from there with deep learning algorithms, combine these radiomics features with other data modalities, and with that be able to demonstrate that we could predict response to some type of immunotherapy with 80% AUC, which is kind of a best-in-class score in the medical field, while still giving to the user the ability to understand what are the features that are defining whether the patient is a good responder or not. Okay? So that's a methodology that I would say is still early. In our case, we use cross-network validation techniques, which are not very, very familiar as well in the pharma industry. So it's not necessarily always easy to educate pharma on those powerful statistical models so that they are then being adopted in the clinical trials and they are being deployed post-approval of drugs. But I think that's definitively the future of the medicine. And for us, being already in 800 centers around the world, computing genomic data, adding this data layer is our next mission to be able to bring even smarter insights to the hospitals that we're serving. We've been demonstrating that this approach works in some type of cancer types. Again, one has to really understand well the biology behind it to be able to solve the problems. And today I'm going to highlight you one case in kidney cancer. It's a collaboration we've made with a hospital in Bordeaux. You all know Bordeaux for the wine, but they do other good things as well. And this in this University Hospital of Bordeaux, they've been working with us to be able to understand what are the risks of different patients in kidney cancer and build these capabilities to serve them for immunotherapy treatment.
Tu pourrais nous décrire un petit peu Europredict, ce que ça fait et
oui,
qu'est-ce que
donc,
tu y vois
effectivement,
derrière?
c'était le sujet de ma thèse de médecine. L'idée initiale, c'était d'arriver à mieux caractériser le risque de récidive après chirurgie d'un cancer du rein. Donc des tumeurs localisées au rein qu'on opère. Un patient qui est considéré comme guéri ou en rémission après la chirurgie, et essayer de mieux déterminer ce qui va faire qu'il y a un patient récidive ou pas. Dans EuroCCR, on a beaucoup de données qui encadrent ces chirurgies du cancer du rein. Des données cliniques, biologiques, d'imagerie, de chirurgie, de suivi, qu'on n'était pas capable, nous, avec des statistiques traditionnelles, d'exploiter pour arriver à mieux caractériser ce risque de récidive. Et donc, l'objectif, c'était de créer un nouveau score de machine learning, un outil pour prédire la récidive chez ces patients.
Ce d'autant qu'en plus, on est chaîné au SNDS, donc le Système national des données de santé. Et ça représente aujourd'hui pas loin de 10 millions de données qui sont à disposition sur des patients pris en charge de façon contemporaine par des équipes françaises pour un cancer du rein. C'est pas tellement une vision, mais c'est finalement un besoin que d'avoir de nouvelles modalités statistiques pour traiter cette donnée et essayer d'en tirer des informations pour améliorer notre capacité à offrir aux patients une prise en charge qui soit le plus personnalisée possible et la plus individualisée.
Et donc l'outil Europredict que nous avons co-développé, Gaël, durant ta thèse, il est déployé, maintenant il est accessible. Quel usage tu en fais dans ton
C'est un outil qui n'est pas encore un dispositif médical certifié, donc c'est pas un outil dont on peut se servir pour changer les prises en charge des patients. Mais par contre, il est très utile pour discuter avec le patient. On peut lui présenter comme un outil de recherche qui nous permet de mieux apprécier son risque de récidive et montrer concrètement au patient ses facteurs de risque de récidive et son risque personnalisé. Et grâce à ça, développer la discussion autour de la surveillance ou d'un éventuel traitement complémentaire à la chirurgie pour la suite de sa prise en charge.
So with that, we arrive to the end of my talk. I don't know if I see happy or sad faces. My point was not, it's all dark, okay? The future is bright. We will all be personally or indirectly affected by these disorders. So for the one who are in the tech world, I hope this inspired you and continue what you are doing so that we can leverage on your tools to build this world of data-driven medicine. For the one who are in healthcare, please reach out. We are very, very in favor of collaborative partnerships. But all in all, let's make this happen together so that people around the world can benefit from the use case of Gabriel and others. With that, thank you very much and have a good day.
Thank you all very much for attending the session today. Unfortunately, that's all the time we have.
Thank you for joining this session.
Thank you
Please
to
remember
Jurgi
fill out the
for
session
speaking
survey
and
at
enjoy the
GTC
rest of your
for a chance
GTC.
to win a $50 gift